Hello, everyone, and welcome to today's webinar, Assurance of Sterility and Sterility Assurance Levels, What's in the Future, brought to you by Nelson Laboratories in partnership with QMed. I'm Tanya von Grumkow, Program Manager at UVM Americas, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. Each widget within the dock at the bottom of the console will open in a different window for you to view additional resources, speaker bios, and more. There are several social media widgets for you to like, share, and talk about the content with your peers and communities. At the end of the program, we will ask you to complete a short survey. Please take a moment to fill this out. Your feedback will provide us with valuable information on the subjects covered in this webinar and how we can improve future events. You can also launch the survey at any time by clicking on the survey button at the bottom of the console. The slides will advance automatically throughout the event. You may also download a copy of the slides by clicking on the PDF label to download slides located in the additional resources widget. If you're experiencing any technical problems, please visit our webinar help guide by clicking on the Help widget. Now on to the presentation. Discussing today's topic will be Martel Winters, Senior Scientist, and Dr. Wendy Wangsgaard, Senior Scientist as well at Nelson Laboratories. Wendy, I now hand it off to you.
Thank you, Tanya, and welcome everyone to our webinar. As Tanya indicated, today we will be talking about the assurance of sterility and sterility assurance levels and what's in the future. So a little background on why we believe this webinar is of importance right now. Currently, we have a document in the U.S., and that's that first bullet point there. It's AAMI ST67, and it talks about the sterilization of healthcare products and the requirements and guidance for selecting a sterility assurance level for products that are labeled sterile. Now, in our industry, within AAMI, we have a working group, and a few years back, we made a decision that a sterility assurance level probably wasn't exactly the idea we wanted to present to the world. And so we collectively decided to change our name to Assurance of Sterility because we felt that better represented and encompassed what we are trying to accomplish with this document. So right now, there's an initiative in the ISO technical committees on sterilization of healthcare products to develop a standard which will discuss assurance of sterility. Now, the content of ST67 will be adapted into this ISO standard and represent current and future industry thinking worldwide. Several topics have been discussed in great detail, which are of interest to the sterilization industry in general. Now, they have put together a new work item proposal, and the idea behind this is it's going to be a risk-based approach. And some of the things that we feel are important to take into consideration as we take this approach is, what is the risk to the patient and or the benefit of a medical device that might be used. Also, the advent of material compatibility constraints is becoming more of an issue. We now see medical devices that are combined with pharmaceuticals or biological materials, and sometimes that can pose problems when talking about a terminal sterilization process. And in association with that, an assessment of the potential of terminal sterilization methods for some of these combination products. Also, to be addressed would be the intended use of the product. Does it have patient contact versus non-patient contact, or direct versus indirect contact? So all of these concepts will be further addressed in an ISO document as opposed to just an AAMI document. Now, taking that one step further, we are in the process of updating ST67. And some of the things that we are going to look at in that document is a continuation of that risk-based approach, including the likelihood of a risk occurring, the degree of harm that might result from that risk, and also what is known about the risk or what ought to be known about that risk, either by the patient, the doctor, the medical device manufacturer, concepts like that. And then also the availability and suitability of ways to eliminate or minimize any risk that might be associated. Now, one thing that we are adding to this document is some things that you need to evaluate when you are looking at the risk. And in this case, commercially available alternative materials that may be compatible with applicable sterilization methods. And what is meant by that is that a lot of work has been done on materials that already exist, and it's known how they react to your common sterilization modalities such as radiation or ethylene oxide, steam, dry heat, those types of things. So there is quite an assortment of information out there that can be used when you are starting to look at how a material might be used appropriately in a new medical device. And then also design features that are compatible with the applicable sterilization methods, such as those that I just mentioned. And, of course, with combination products coming up and the sensitivity of some of these materials, there might be alternative validation processes that might be more appropriate for something that might not be able to handle some of these terminal sterilization processes. And then, of course, other sterilization methods, which will be discussed a little bit more detail by Martel. Now, the slide here is just kind of an overview of the standards that are out there right now, and also what we are relying heavily on as we put these documents together. EN 556 is the European standard that is comparable to AAMI ST67. However, in that document right now, a product is not able to be labeled sterile unless it actually meets a 10 to the minus 6 sterility assurance level. And that is something we will be addressing with different approaches in the upcoming documents. We will be also relying heavily on standards that are already in existence. ISO 14971 specifically discusses risk management, and so we will refer heavily to that document and the guidance that is provided there. We are also working in conjunction with PDA, and they have many technical reports that are available that discuss their approaches. We will be pointing to those, and then also working very closely with the FDA, and they have guidance documents out there. So we will be pointing to all of these type of standards for additional guidance, and they are helping provide the backbone of what we are putting together right now. And this has already been mentioned. It was in the title, but we can't go anywhere without talking about sterility assurance levels. And the definition of that is the probability of a viable microorganism occurring on an item after sterilization. Now, it's good to note that an SAL is normally expressed as a 10 to the minus number. And the example that we have down below is that it is a quantitative value. So common industry is that you are sterilizing to a sterility assurance level of 10 to the minus 6, but that number is actually lower than a 10 to the minus 3. So that is a concept that you need to be aware of. When it comes to sterility assurance levels, it is widely used in the application of a terminal sterilization process. And the medical device industry has defaulted to using the term sterility assurance level when talking about a sterile product. A concept that we are going to introduce in the new document is actually probability of a non-sterile unit. This is the term that is typically used in the pharmaceutical industry. And you are talking about the probability of a non-sterile unit occurring. And for the purposes of this document, it is going to be synonymous with a sterility assurance level. and it is also expressed in a 10 to the minus number. Now, sterility assurance level, it's been used in a variety of ways, and not always correctly so. And so we are going to try to provide some guidance on when it should be used, and it will point towards when you are using a terminal sterilization process, that would be expected. When we're talking about aseptic processing, sterility assurance level should not be applied. That's what this reinforces right here. Don't do it. I am now going to turn the time over to Martel Winters, and he will go through some examples and a bit more detail about some of the concepts that I have introduced here. Thank you for your time, and here's Martel.
Thank you, Wendy. So to start off with an example here regarding sterility assurance levels and when it is appropriate versus not appropriate to apply that term. This first example, so a fractional exposure of a sterilization process is applied, and 100 samples undergo a test of sterility, and in that test, one of them shows microbial growth. So what we've actually proven a 1 in 100 contamination rate with data. And keep in mind also that a bioburden reduction step has been applied, and this is to the finished product, and which means that it's also in its final packaging. So in this situation, it is appropriate to call this contamination rate of 1 in 100 an SAL of 10 to the minus 2. And also, since it's appropriate to call it an SAL, then these data can also be used to extrapolate or to determine a cycle that would be an SAL of 10 to the minus 6, which is always a longer cycle, longer exposure. Now, with terminal sterilization, just to make sure on the same page regarding definition, that a product is sterilized after being sealed in its final package. And anytime that we are talking about terminal sterilization, there is always associated with that a particular sterility assurance level which has been determined. And also, in validation of that sterilization process, we are always doing some type of fractional or half cycle. For those that are more visually oriented, here's an example of what we might call a death curve. So this is a situation where we are starting off with 1 million microorganisms, 1 million CFU of microorganisms, and on the bottom axis, as time or as dose increases, the blue line decreases. So microorganisms are killed with longer exposures to the sterilization process. We are usually performing testing somewhere in the middle of that range, of that death curve. So we're usually doing testing after some degree of exposure in the 10 to the 0, 10 to the minus 1, 10 to the minus 2 range in that area. And then after the testing is performed at that point, then we are doing the terminal sterilization at a longer exposure, or which would equate to a 10 to the minus 6 cycle. Now, when we're talking aseptic processing, it's very different. Here we are sterilizing the components as in their own bags or containers, and then we are assembling all of the components in an aseptic manner. And this type of process is always validated using what's called a process simulation. It's also known as a media fill. And in a media fill, the containers where they are usually filled with the product, instead they are filled with growth media. And then once the containers are capped or sealed, then they are incubated and for a period of time, and then it is determined if there's any growth in any of the containers. So if we think of it from an aseptic processing standpoint, here would be an incorrect example of application of sterility assurance level. So after a media fill in an aseptic process, 1,000 containers are filled with media, and products are tested for sterility. And if one of them demonstrates microbial growth, so we have proven with data a 1 in 1,000 contamination rate. So unfortunately, sometimes people start to make the connection here with SAL, where they say, well, 1 in 1,000 is the same as 10 to the minus 3. And some people might use that terminology, start to employ the SAL concept. But in actuality, 1 in 100 or 1 in 1,000 contamination rate does not always mean 10 to the minus 2 or 10 to the minus 3. And the question that we ask is, has a bioburden reduction step been applied to the finished product? And that's a critical question to ask, because as part of the whole concept of SAL is an understanding that we are applying a shorter cycle and obtaining data at a shorter cycle, and then using those data to substantiate a longer cycle, which is used for terminal sterilization or complete sterilization of the products that are sold. That means that to truly have an SAL, that there is some type of active bioburden reduction step which is being applied to the finished product. So with aseptic processing, as I mentioned, the components are sterilized ahead of time, and the components usually are sterilized to 10 to the minus 6 SAL, and then they're assembled in an aseptic manner and as clean of an environment as possible. But in this situation, there is no bioburden reduction step being applied to the finished product. There is no terminal sterilization. The only sterilization that is being applied is in different forms to the component. So the real point here is that you cannot lengthen the aseptic process or lengthen anything in the process or afterwards, which helps you to obtain a greater assurance of sterility. And that is the primary distinguishing factor between the use of SAL for terminally sterilized products versus the idea that we do not use SAL for aseptic processing. So then the next question is, well, for aseptic processing, what is our target? What value do we apply to aseptic processing? And the answer to that is that we actually treat aseptic processing very differently. In terminal sterilization, there is always a target sterilization level, and it is nearly always 10 to the minus 6. For aseptic processing, rather than having some type of target level, the approach is validation of the process. A target level for aseptic processing would require that every batch manufactured have a certain quantity of products in that batch, and batch sizes vary widely from company to company and from product to product. So to try and establish a minimum required quantity of products to be tested so you can prove a particular contamination rate is not appropriate. So the validation usually consists of testing 3 lots of the worst case batch of that product, and either performing a media fill or if it cannot be filled with media, then the process simulation terminology is more applicable, and it follows the same concept. And under that approach, you are validating the process, and then having very good process controls and process monitors in place to ensure that the manufacturing process is just as good today as it was yesterday when we performed our media fill or the process simulation. So we want to talk a little bit about the SAL 10 to the minus 6 number. The question I frequently ask is, upon what plethora of data is the 10 to the minus 6 requirement based? And when you dig into the history behind that, it was a completely arbitrarily established number. 10 to the minus 6 corresponds to a 1 in a million probability of a non-sterile product, and 1 in a million sounds like a great number, but there really is no data or no testing behind that to show that 10 to the minus 6 is the required number. So then if we start thinking what really is required for patient safety, and we've found in the last few years that it really depends on the situation. It can be as high as 10 to the minus 3, 10 to the minus 4, or 10 to the minus 5, rather than 10 to the minus 6. And there are a couple of links there to 2 different papers, and both of which are related to sterility assurance level and patient safety or patient infection. And the concept behind both of those is to using a mathematical model to determine what SAL for that specific product truly is necessary for the to ensure that the product is not increasing the potential harm to a patient. Now, it should be noted that there are cases where 10 to the minus 6 probably is necessary. So we will likely not ever see a situation where the overall expectation is changed to 10 to the minus 3 or 10 to the minus 4, for example. So we should not expect to change the target SAL from 10 to the minus 6 to something else. But in an effort to provide a structure for some of these new innovative combination devices, medical devices, biological products, and tissues, then we want to provide something to where where manufacturers have something to go to rather than having to default automatically to aseptic processing. The approach that is currently in ST67, and largely this same approach will be kept in the new version, and we anticipate that this same general flow will also be used in the ISO document, is first to select 10 to the minus 6, and then determine if the product can handle that. And if the product for some reason cannot take a 10 to the minus 6 process, then verify that that is truly the case. Can it truly not take any form of 10 to the minus 6? And Wendy already mentioned ideas of alternate sterilization methods or alternate validation approaches. For example, rather than the typical overkill type method in an ethylene oxide cycle, then might consider a more optimized method like a BI bioburden or a bioburden-based method, which can really shorten those cycles. Now, if it truly cannot take any form of 10 to the minus 6, then perform the risk assessment, which Wendy also mentioned, to help understand which SAL is really necessary for patient safety for that specific product and the use of that product. When addressing use of an alternate SAL, there are a few things to consider. So and one question that is frequently asked is how much data do you have to have in place in order to select an alternate SAL? For example, must every sterilization method known to man or ever hypothesized or potentially discussed in an article or something, must every single thing be attempted before you can accurately say it can't handle 10 to the minus 6? And the answer to that question is no. As we've been working on the improvements, the update to ST67, and the approach that we intend to put forward in the ISO document, the consensus truly is to demonstrate a reasonable attempt at using the various sterilization methods. And the term that we are using in this new version of ST67 is reasonably practicable, which is kind of a mouthful, but is meant to get across the idea that go through a reasonable attempt at showing that other sterilization methods or sterilization validation approaches are not feasible. Then, as you go through the risk assessment, you will find that it can help shed light regarding how much evidence really is necessary. As you're determining which SAL is appropriate, you can also get a feel for how hard you need to try these other methods or validation approaches. So you see the risk assessment really does play a critical role in this discussion of selection of an alternate SAL. So as we dig into what is reasonable or what is reasonably practicable, here's kind of an example. The first step would be if there is obvious scientific literature or obvious understanding in the industry regarding something in particular, then that is something that certainly should be considered. For example, if your product is inside, it must be inside a foil pouch or inside glass, it is obvious that ethylene oxide cannot be used to sterilize those. That is well understood. So then the next you might find, is there can a product withstand the typical sterilization processes? So for radiation, for example, you might try a 15 and a 25 kilogram dose of radiation. The 25 kilogram dose is a very common dose for most products, and 15 kilograms is on the lower end of a sterilization dose that could be used, and determine if there is anything there as far as damage to product, and if you see a significant decrease between 25 and 15, then you may go on and try, say, an 8 or a 10 kilogram dose and just see if there's a level at which is perfectly acceptable. And taking the same approach with the typical sterilization type is expected: ethylene oxide, radiation, steam, dry heat. Then the next step will be to gather some empirical data on process optimization. So, for example, if we stick with the radiation example, if I try 15 kilogram and get decent results, try, say, a 12 kilogram dose and get better results, then I should also look at other things I can optimize. For radiation, for example, typical ways to decrease damage to product would be to irradiate at low temperatures, or to purge the oxygen from the package before sealing. And then, as I mentioned previously, looking at a bioburden-based method rather than overkill, which is radiation is always a bioburden-based method, but within radiation even there are other methods there than the more traditional validation approaches that can be used to result in lower doses. And the same would be expected for if it appears, for example, that ethylene oxide is showing reasonable results, then the next expectation, if you need something shorter, would be to look at a BI bioburden or a bioburden-based approach. If we compare the two, so we're talking aseptic processing versus SAL, rather than compare, with both of them, really there needs to be some type of rationale for going to aseptic processing, just like we talked about with an alternate sterility assurance level for terminal sterilization. So for aseptic processing, there might be a tendency to say, well, 10 to the minus 6 isn't going to work. It's not going to be easy. Let's just go to aseptic processing. And it can be easy to think that until you begin to understand what is truly involved in aseptic processing. And just as we talked about having a good rationale with good data behind it to select an alternate SAL, the expectation with FDA, clearly in our discussions, is that there should also be a rationale in place for selecting aseptic processing for a product which really should have a sterility assurance level. And aseptic processing is not an easy answer. There is a lot involved with setting up an aseptic process. There are greatly increased microbiology process controls. The equipment and the rooms are more expensive and require a lot more maintenance, a lot more extensive validation and routine monitoring in those rooms and with the equipment as well. So it is certainly not something that should be leaned on as an easy out to terminal sterilization. Some things that you might address or include in a rationale to go with aseptic processing. I think the first expectation would be that there would be data to show that the only cycles which result in functional product, with those cycles there is too much residual risk to the patient. For example, if the only possible sterilization cycle that can be determined using the best sterilization method for the product results in SAL 10 to the minus 1 or 10 to the 0, for example, then that probably could be considered as too much potential risk to the patient. And an assessment also of other alternate materials, for example, switching out one polymer for another, that would certainly be an expectation if that could be done. Showing that both typical sterilization cycles don't work and that optimized sterilization cycles do not work, meaning a bioburden-based approach. So there's an idea of things that should be expected in a rationale for going into aseptic processing. The next question then is, can the product truly be aseptically manufactured or assembled? There are some products that might be fairly difficult to assemble, and you might discover that as part of the risk assessment that in order to assemble this product, there is so much manipulation that even an SAL 10⁻⁻1 might be better than the aseptic process just because of the difficulty. And another option is components which have living cells. There is a separate ISO document which is just about to be published. It's 18362. So certainly if the product requires living cells, then they cannot be sterilized, and all the other components of the product can be, but one component of that product, which would be the living cells, would not be sterilized prior to the aseptic assembly or aseptic process. So those are things to consider. So when looking at an alternative SAL or aseptic processing, in either case, again, the risk assessment is critical. It plays a critical role. And the risk assessment must be very detailed and very complete. And it's critical to have knowledgeable people involved. There are often people in even smaller medical device companies that are very familiar with risk assessments. But this type of risk assessment does not involve only the manufacturing aspect, the sterilization aspect, the impact of product from sterilization. A deep knowledge of sterilization processes and how those can be optimized is critical. And that type of knowledge is often not present internally. So please involve someone who does have that deep knowledge on the sterilization side as well as part of that risk assessment. So the nice thing about all this is we're in a very fun time right now in the healthcare industry. There's some exciting things going on, wonderful new products coming up, and as I mentioned, the primary goal behind trying to establish these approaches is to provide some background, some structure to companies that have these exciting new products which cannot undergo a traditional sterilization process. And that is the intent, and we hope that in providing this, that will allow manufacturers to indeed come up with even more new exciting products for the healthcare industry. And I turn the time now back over to Tanya.
Thank you, Martel and Wendy, for providing the valuable information on assurance of the sterility and sterility assurance levels. For upcoming webinars and for the archive of this webinar, please visit qmed.com. This webinar is copyright 2015 by QMed. The presentation materials are owned by or copyrighted by Nelson Laboratories. The individual speakers are solely responsible for their content and opinions. On behalf of QMed and our sponsor, Nelson Laboratories, thank you for attending and have a great rest of your day.